Welcome to this explainer. Today we're going to explore something absolutely mind-blowing. We're looking at the actual instruction manual that builds every single one of us. We are decoding the fascinating science of human genetics and breaking down the exact mechanisms that make you, well, you.
Think about it for a second. What exactly dictates your specific height or the exact color of your eyes? How does one tiny microscopic cell know exactly how to build a complex human being? Well, the answer is hiding right there, deep inside your cells.
And it all boils down to a surprisingly specific number, roughly 30,000. That's the approximate number of genes contained within the entire human genome. It's an unbelievable sequence of data. And crazy enough, we only published the very first rough draft of this sequence back in 2001.
Okay, Section 1, the blueprint of life. Let's dive right into our cellular foundations.
Deep inside the nucleus of your cells, you've got DNA tightly packed into chromosomes. It's this beautiful double helix structure, and it is literally the physical carrier of your genetic code. Think of it like a giant, coiled-up biological hard drive sitting right there inside the nucleus, holding all the instructions for everything you are.
Now, a gene is basically just an independent unit on that DNA strand. These individual genes carry the code that dictates the production of essential enzymes and molecules which run your entire body. But here's the catch. Actually, this is a really important distinction. They are only active when they're unwound in a strand form, known as chromatin. When they're tightly packed up into that compact chromosome form, they completely shut down and become inactive, just waiting for the exact right moment to be read.
If we zoom out to a macro view inside your cells, we find the specialized X and Y chromosomes. These are the sex chromosomes, and they determine biological sex. Females typically have 2 X chromosomes, and males have 1 X and 1 Y chromosome.
All right, zooming out a little bit further, we each possess exactly 46 chromosomes inside our cellular nucleus, and they're arranged in pairs. So you have 22 pairs of what we call autosomes. These handle the genes for the vast majority of your traits, plus that one remaining pair of sex chromosomes we just talked about. Put them all together, and bam, those 23 pairs form your complete human genome.
Moving right along to Section 2, the rules of inheritance.
So, how do you get this perfectly unique mix of traits from your parents? Well, it happens during meiotic cell division, which produces the gametes. During this process, chromosomes don't just politely sort themselves out independently, they undergo a literal crossing over of genes with their chromosome partners. It completely shuffles the genetic deck, which creates the incredible genetic variation we see in humans.
Because you get one chromosome from each parent, you inherit 2 versions of every single gene, right? These are called alleles. If your 2 copies are a perfect match, your genotype for that trait is homozygous. But if those 2 alleles differ from one another, you're heterozygous for that gene. So the big question is, what happens when those 2 alleles are different? That's where dominance kicks in. Dominant genes have effects that will absolutely show up in your physical appearance, your phenotype. Recessive genes, on the other hand, get totally masked by a dominant partner. They don't visibly appear, meaning they just hide in the background, making you a silent genetic carrier for that specific trait.
It's a literal game of chance. Let's take albinism, for example. When a mother and a father are both normal carriers, meaning they each have one dominant normal gene and one recessive gene, there are specific odds at play. Because they're both carriers, there is exactly a 1 in 4 chance, or a 25% probability, that their child will inherit both recessive alleles, which results in the actual physical phenotype for albinism.
Which brings us to Section 3, when the code changes.
We know how the manual is supposed to work, but what happens to the human body when there's a typo in the manual? Well, individual mutant genes can be passed down, and these single gene conditions can be absolutely devastating. Take cystic fibrosis, a recessive condition causing a really dangerous buildup of mucus, or phenylketonuria, or PKU, which causes a toxic accumulation in tissues that can lead to severe brain injury. And then there's Tay-Sachs disease, where the failure to make just one specific subunit of an essential enzyme causes abnormal lipid accumulation in the brain. Each of these profound challenges traces back to literally a single typo in the DNA. But typos don't just happen at the single gene level. Sometimes we see large-scale chromosomal breakage, or a phenomenon called non-disjunction. That's basically when a chromosome pair completely fails to separate properly during gamete formation. And when that happens, offspring can end up with a chromosome triplet, called a trisomy, Or they might be missing a chromosome entirely, which is called a monosomy.
These massive chromosomal typos result in profound developmental changes. For instance, Down syndrome is usually caused by an extra chromosome 21, making it a trisomy 21. Klinefelter syndrome happens when a male has 2 or more X chromosomes, resulting in an XXY combination. On the flip side, Turner syndrome is a monosomy, where a female is missing an X chromosome, simply designated as XO, which can cause cardiovascular defects and learning disorders.
Now here's a massive plot twist. It's not just inherited mutations that change our genetic destiny. Enter epigenetics. This is the science describing how environmental factors actually trigger chemical DNA changes that alter gene activity, all without changing the core sequence. For example, abnormal levels of chemical methyl groups, or methylation, are associated with some cancers, and the overmethylation of part of the X chromosome is literally what causes Fragile X syndrome.
And finally, Section 4, the future of genetic medicine. Since we can read the blueprint and spot the errors now, how is modern medicine empowering us to actually rewrite the code? Well, the first step is identification. Today, genetic counselors use pedigrees to map out family histories and karyotypes to physically spot chromosomal abnormalities. We even use incredible techniques like amniocentesis, collecting fetal cells floating in the amniotic fluid to identify genetic conditions before a baby is even born.
Historically, most treatments just focused on relieving symptoms. But now, we are entering the revolutionary era of gene therapy. This is a total game changer. We're talking about gene replacement therapy, where abnormal genes are literally completely replaced by therapeutic ones. Or gene augmentation therapy, where new cells carrying normal genes are introduced into the body to pump out a much-needed protein. The delivery methods for these therapies are straight out of science fiction, but they're happening right now. Scientists are actually modifying harmless viruses and using them as delivery vectors to drop therapeutic genes right into the body. This is already showing early promise in curing a genetic form of blindness. Plus, researchers are using RNA interference in labs to literally silence specific disease-causing genes, just turning them off one by one.
So, we have completely traversed the microscopic universe of the human genome, from the 30,000 genes that build us, to the rules that pass them down, to the very real tools we now have to alter them. Which leaves us with a pretty profound question to ponder. We now have the growing power to silence the genes that cause disease and relieve immense suffering. But as this technology advances, how else will humanity choose to rewrite the blueprint of life? What comes next in our biological evolution? Thanks so much for tuning into this explainer, and definitely keep exploring.